So let's break down and explain all of Module 1 for GCSE Biology. Now this video is specifically targeted towards the AQA specification. However, there's so much overlap between the specifications that this would also be really suitable and beneficial to absolutely anyone on any exam board. So let's get started. Now, Module 1 is broken up into three main parts. Our first part is cell structure, then we have cell division, and then we have transport in cells. So let's start with our cell structure. Our first example of a cell is the animal cell here. And we're going to go through and label each part of our animal cell. So first we have our nucleus. Now the function of the nucleus is to control the activity of the cell. So control the activity of the cell. Now the way that it does that is by containing the DNA or the genetic material. Our next part of the cell there, we have the mitochondria. Now the mitochondria, these are the site of aerobic respiration. Now in an exam question, it would be really good to get into the habit of being specific in your answers. So if you can say aerobic respiration, then you should say aerobic respiration. So the site of aerobic respiration. And because of the aerobic respiration, the purpose of that is to release energy. We're not going to say produce energy, we are going to say release energy. Our next part of the cell is the ribosomes. Now the ribosomes are these smaller parts of the cell that if we were looking with a light microscope might be quite difficult to see. The ribosomes, these are the site of protein synthesis. This is where proteins like enzymes are made. Our next part of the cell is our cell membrane. Now the cell membrane is this external surface and it controls what enters and exits the cell. Controls what enters and exits the cell. And it does that by being what we would describe as a partially permeable membrane. That means that only some things can pass through it and other things cannot pass through. And our last part of an animal cell there is the cytoplasm. The cytoplasm, this is where the chemical reactions take place, as well as just being the internal part of the cell. So where the chemical reactions happen. Now the chemical reactions of a cell, this is known as the metabolic reactions too. So we could say where the metabolic reactions take place. This is our animal cell. Let's now compare this to a plant cell. So our plant cell has a few structures that are also found in animal cells. This would be the nucleus, the cell membrane, the mitochondria, and the cytoplasm, and not drawn on this diagram would also be the ribosomes. And in addition to these parts of the cell, the plant cell also has some specialist features too. The plant cell has a cell wall. Now the cell wall is made of cellulose, a specific type of carbohydrate. And that cell wall provides structure and support to the cell. Plants don't have a skeleton, it is the cell wall that keeps the plant upright. Structure and support to the cell and the plant as well. Plant cells also have chloroplasts. However, as we will see, not all plant cells have chloroplasts. The chloroplasts are the site of photosynthesis. And the purpose of the photosynthesis is to produce glucose or to produce food for the plant. And then finally, we have a permanent vacuole. The reason we say permanent is because it is always there in the plant cells, whereas in animal cells, sometimes they might have a vacuole, but the vacuole might then disappear as well. Now, the permanent vacuole contains the cell sap. Now, the cell sap is a kind of watery liquid, a watery substance that also contains sugars and salts as well. And what it does is it swells up and expands the vacuole, and that pushes against the cell walls, and that causes pressure inside the cell, keeping the cell kind of taut, maintaining the structure of the cell. We could say that it maintains the turgor pressure, the pressure inside the cell, keeping the shape of the cell. So to maintain the turgor pressure, or we could say the shape of the cell. Let's now look at another type of cell, a prokaryotic cell. So the first two, the animal and the plant cells, were examples of eukaryotic cells. Now we have a prokaryotic cell. A prokaryotic cell means that there is no nucleus, whereas the eukaryotic cells would have a nucleus. An example of a prokaryotic cell would be a bacteria. So a bacterial cell is a prokaryote. Let's go through and label our different parts of the prokaryote because there are some differences here. Our first part here, A, this is our cilia. B is what we call a plasmid. Now, a plasmid is a small ring of DNA that can be transferred between cells. C is a loop of DNA. Notice how that loop of DNA is free in the cytoplasm. It is not found within a nucleus. D is the cytoplasm. All cells will contain a cytoplasm because the cytoplasm is just the inside of the cell. E is the ribosome. My diagram here is very much not to scale. The plasmid would be significantly smaller than the ribosomes in reality. F is a flagellum. Some people might refer to this as a tail, but technically it is a flagellum. It is a structure of the cell that helps the cell to move. G is a slime capsule. The slime capsule helps to protect the cell as well and helps to protect it from certain chemicals. H is the cell wall. The cell wall of the prokaryote is slightly different to the cell wall of the plant cell, but still helps to maintain the shape of the cell. And then I is the cell membrane. So every single cell will contain a cytoplasm and the cell membrane because the cell membrane is what makes it a cell. Now, not all of these parts of the cell are actually found in every single prokaryote. Not all prokaryotes will have the cilia, not all of them will have the flagellum or the slime capsule either. Some of them you may not see these structures on the surface of the cell. So those are the general structures of animal cells, plant cells, and prokaryotes. But we also need to know some specialized cells as well. Now, these specialized cells have a slightly different structure, and the structure relates to their function. And in an exam, it's really important that you link the structure to the function. So let's break it down. Our first example of a specialized plant cell is a palisade cell. And the function of the palisade cell is to carry out photosynthesis. They would be found in the leaf, the main photosynthetic organ of the plant, and they are there to capture as much light as possible for the photosynthesis. So the adaptations of the palisade cells, the first one is that they have many chloroplasts. And by having many chloroplasts, it is more likely that the light is going to hit one of the chloroplasts, and that means that they are going to produce more glucose. They're going to have a faster rate of photosynthesis. They are also quite long and thin and form these column shapes. And again, that is to maximize light absorption. So to maximize the light absorption. Our next type of plant cell is a root hair cell. Now, the root hair cells are found on the roots underground, and the purpose of the root hair cells is to absorb the water and the ions from the soil. So how is the root hair cell adapted for this function? Well, the first part is it has this root hair, this long, thin structure coming out from the cell. The root hair increases the surface area. And by increasing the surface area, what that does is that maximizes the rate of transport in and out of the cell. So it maximizes the rate of transport in and out of the cell. The root hair cell, we would also expect to find many mitochondria within the root hair cell too. The reason why we would expect to find many mitochondria is because the root hair cell might be doing a lot of active transport, and those mitochondria are going to be releasing the energy for the active transport. An additional adaptation of the root hair cell is that it does not contain any chloroplasts. It is underground, which means there's no sunlight, so there would be no point in the root hair cell producing these chloroplasts. So to save energy, it doesn't bother. Our next type of specialized plant cell are the xylem vessels. Now the xylem vessels, they form part of the xylem tissue, and this is used to transport water up the plant in the transpiration stream. Now, the way that the xylem vessels are adapted for this is that they are dead cells with no end walls or organelles either. They also have no organelles. The purpose for this, the reason for this structural adaptation, is that this means that we have a continuous column of water going all the way up the plant. So a continuous column of water up the plant, and that enables transpiration to take place. Another adaptation of the xylem vessels is this compound here called lignin. Now, lignin supports the xylem vessels and helps to support the plant as a whole. It stops the xylem vessels from collapsing and closing. The xylem is a little bit like a straw, right the way from the leaves to the roots. Let's have a look at our specialized animal cells then. Our first example of a specialized animal cell is the sperm cell. Now, the function of the sperm cell is to swim to the egg. And once it gets to the egg, it needs to fertilize it. So how is it adapted for this? Well, our first adaptation is the tail. The tail enables the sperm cell to move. Some people refer to the tail as a flagellum, and technically this is not a flagellum. The tail enables the sperm cell to move. We also have the midpiece with many mitochondria. And the reason that there are many mitochondria, again, is to carry out more respiration to release energy for movement. In the head of the sperm cell, we have a nucleus, but the nucleus of the sperm cell is adapted as well. We say that it has a haploid nucleus. If you're not sure what that word haploid means, go check out my other videos where I break down and explain meiosis. Now, the haploid nucleus, that enables the sperm cell to fertilize the egg. The nuclei fuse together to form a diploid nucleus. And then finally, at the very, very tip of the sperm cell, we have the acrosome. And the acrosome contains enzymes. It contains enzymes that will break down the surface of the egg cell, allowing the sperm cell to plant its nucleus into the egg cell. That break down the surface of the egg cell. Our next specialized animal cell is a nerve cell. Now, a nerve cell is adapted to carry electrical impulses around the body. Those nerve cells, they are very, very long to carry the impulses a long way. They are also highly branched, and that enables them to connect to other nerve cells. As well as that, they have this fatty sheath that helps to insulate them. They have a fatty sheath, and that enables the electrical insulation. Our next type of animal cell is a muscle cell. Now, the muscle cells, the function of the muscle cells is to contract to produce movement. So how are those muscle cells adapted? Well, they are long, and that enables them to contract. They also have many mitochondria. And that enables them to release energy for movement. As well as having many mitochondria, they also are going to have many ribosomes. And thinking back to the function of the ribosome, that is necessary for protein synthesis. When we talk about needing protein to build muscle mass, the ribosomes help with that. There's two more examples of specialized animal cells we need to know about. This one on the left here, we have a red blood cell. Now, the red blood cell, the function is to transport oxygen around the body. And it does that as part of the blood. To help it do this, the red blood cell contains hemoglobin. Hemoglobin is an example of a protein that binds to oxygen. It is also red, and that's what makes the red blood cells red. The red blood cell also has no nucleus. The nucleus gets removed as that red blood cell develops into its specialized structure. Now, by having no nucleus, this makes more space for the hemoglobin. And we could say that it has a biconcave shape. Both sides of the red blood cell dip in like this. So a biconcave shape. And that biconcave shape, what that does is increases the surface area to volume ratio. And this maximizes the amount of oxygen that diffuses into the cell. Maximizes the amount of oxygen that diffuses into the cell. Now, our last type of cell here is involved in absorption and digestion. So we can perhaps make some predictions as to what adaptations there would be. Well, if it's involved in absorption, well, perhaps it might have cilia or microvilli as well. Microvilli are the folds of the cell membrane, and this increases the surface area. It might have the cilia to move the extracellular fluid, to move the fluid outside the cell. The reason why that would be a benefit is because it moves and replaces the fluid outside the cell with fluid that has a higher concentration of things like glucose, and that would increase the rate of diffusion into the cell. If it's involved in digestion, it's probably going to be producing enzymes, which means it's going to have lots of ribosomes. Lots of ribosomes to produce enzymes. And if it's doing lots of absorption, it might be doing lots of active transport, in which case it would have lots of mitochondria as well. In an exam, you might be told about a cell that you don't know about and be told some of the functions of that cell. And based on those functions, you might be asked to suggest what adaptations it would have. So if it's carrying out movement, it would probably have lots of mitochondria to release the energy. If it's producing proteins, it will probably have lots of ribosomes. You need to be aware of all of these different structural adaptations and how they link to the function of the cell. This cell is called an epithelial cell on the right-hand side. So cells are microscopic. They are too small for us to be able to see with the naked eye. To be able to see those cells, we need to use a microscope. Let's break down the different parts of a light microscope. So at the top here, we have the eyepiece lens. This is where we look into the microscope. Underneath, we have the objective lens. This is the piece just above the slide. The slide would go here onto the stage. And underneath the stage, we would have a light source. That could be a bulb or perhaps a mirror that reflects the light through the slide. We also have two different focus wheels. We have a coarse focus wheel and we have a fine focus wheel. And we will get onto the difference of those two different focus wheels in a minute. Now, when we think about our microscope, there are two main terms that we think about: magnification and resolution. The magnification is how many times larger the image is compared to the actual size. So for example, if I had a picture of a cell that was 10 centimeters across, well, that's many, 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 many times bigger than the actual size of a cell, which may be about 1/100th of a millimeter across. What about our resolution? Well, the resolution is the ability to distinguish between two points. So what does that mean? Well, imagine two very, very small parts of a cell. For example, a ribosome. A microscope with poor resolution would mean that we weren't able to make out the difference between those ribosomes, or perhaps make out the edge of the ribosomes. Or maybe, if we were talking about the mitochondria, we perhaps wouldn't be able to see the internal structures of the mitochondria. We wouldn't be able to determine that detail, no matter how well focused the image was. You can think of resolution a little bit like the resolution of a TV. You can have an incredibly low resolution TV and the picture looks pretty rubbish. You can't make out the fine detail on somebody's face. Whereas you could have much, much higher resolution TV, like a 4K resolution. That means you're able to have much more detail in the image and able to see those differences. So how do we correct the images produced by a microscope? What if we have a blurry image? Well, a blurry image is because it is out of focus. So the blurry image, we need to use the focus wheels. We use the focus wheels to focus the image. The coarse focus wheel moves the entire stage up and down and is for large scale adjustment. The fine focus wheel has a much smaller range of movement within the lens itself, and that's for getting the kind of perfect clarity. This makes the image clearer. What about if the image was too small, or the cells in the image were too small? Well, if the cells in the image are too small, we need to increase the magnification. We are not going to say zoom in. You will never get marks for saying zoom in. We say we increase the magnification. And the way that we could increase the magnification is we could switch to a higher power objective lens. What about if we can't make out the finer details? What about if we can't make out the mitochondria? So we can't distinguish the mitochondria. Well, the problem there is that our resolution is too low. So we would need to use a different microscope with a higher resolution. Now, to use a microscope with a higher resolution, we would need to perhaps switch to an electron microscope, which uses a beam of electrons instead of visible light to be able to produce an image. Now, there are some advantages and disadvantages of light and electron microscopes. Well, one of the big advantages of a light microscope is that we can use a living sample. We could look at the movement and the behavior of a living sample, of a cell, perhaps some invertebrates as well. An electron microscope, the disadvantage is it needs a vacuum to be able to function, and so the sample must be dead. We can't view a living sample with an electron microscope. But the big advantages of the electron microscope are that they produce higher magnification and resolution. The disadvantage of the light microscope is that they have much lower magnification and resolution. Now, a common answer that students give for a question like this is they say that electron microscopes are black and white, which is a disadvantage. Now, unfortunately, that doesn't reliably get you marks in exams because the images produced by an electron microscope can be false coloured. We can add colour to them later. So avoid saying that the image is black and white or that the image is in colour for light microscopes because you might not get the marks in your exam. Let's talk through how we use a microscope to view a slide then. Well, first we need to take our specimen and place it onto the slide. The specimen, when we prepare our specimen, we want to cut it to make sure that it is thin. That is so that the light can pass through the specimen. We are then also going to add a stain to our specimen. An example of a stain that you might have used in school is iodine. Iodine binds to the nucleus and shows us the nucleus specifically. So the stain now enables us to see specific organelles. We're then going to place a cover slip over your specimen, lowering it slowly and at an angle, and that is going to prevent air bubbles. You might have done this in school and you might have not done it very, very well, and that means you might have got air bubbles onto your slide, and the air bubbles then distort the image and make it harder for us to see the cells clearly. We're then going to clip the slide onto the stage and start by using the lowest magnification objective lens. The reason for that is because that produces the largest field of view. It means we can see the most number of cells initially, and we can then determine where we might want to produce a larger image and therefore increase the magnification. We're going to look down the eyepiece lens and focus the microscope using the coarse focus wheel. We can then improve the focus using the fine focus wheel as well. When we've focused it on the lowest magnification, we can increase the magnification by using a higher powered objective lens. This will then make the image blurry, so we would then need to focus again. At high power objective lenses, we would only use the fine focus wheels because the coarse focus wheel moves the whole slide up and down. And if we're moving the whole slide up and down, there is a risk where we could damage the slide or potentially the microscope. So we would want to use the fine focus wheel. Now we need to be able to work out what the total magnification of the microscope is. We are using two different lenses to do this. The total magnification of a microscope is equal to the magnification of the eyepiece lens multiplied by the magnification of the objective lens. So, for example, if we had an eyepiece lens that was times 10 and an objective lens that was times 20, the total magnification would be times 200. Another part of the microscopy required practical is understanding how to calculate the image size, the actual size, or the magnification from an image and some other data that has been given to you. So the real length from point X to point Y is 0.06 millimeters. Now the image length we measure on the image itself. So I would get a ruler out, I would measure this, and when I measured it earlier, I measured it out to be 3 centimeters. And we are asked to calculate the magnification. Now for our calculation questions, I'm always going to use the same method, which is ICES. And I always put 3 S's at the end. Our I stands for our information. So the first thing I'm going to write down is that my real length, or my actual size, is equal to 0.06 millimeters. My image length is equal to 3 centimeters. And my magnification, I don't know. That's what I'm trying to calculate. Next, my C is for my conversion. So I'm going to look at my data and see, have I got it in the same values? No, I don't. My image is in centimeters, my actual size is in millimeters. I need to make sure that they are in the same value. So I'm going to convert my centimeters into millimeters. To do that, I'm going to multiply by 10. So we get 30 millimeters. Next is our equation. And our equation is one that you need to learn. Image size equals our actual size times the magnification. You need to learn this equation. It will not be given to you in the exam. Then I substitute in my values into my equation. So my image size is 30, my actual size is 0.06, and my magnification is just m because I don't know it yet. And then our second S is to solve our equation. So I'm going to divide both sides by 0.06. M equals 30 divided by 0.06. And then our final answer, we get 500. Now the final S here is for our significant figures. So if the question says to us, give your answer to 2 significant figures or 3 significant figures, depending on the question, I would need to make sure I give my answer to the correct number of significant figures. This is a really, really common area where students lose marks in an exam, and it's really, really easy. So we want to make sure we're getting that correct. Now in this question, it doesn't say that, and my answer is to 1 significant figure anyway. Let's have a look at another example. So the image in the second picture has been magnified by times 500. calculate the real length of the embryo. Give your answer in micrometers. So when I measured it earlier, I measured the value of x to be 4.1 centimeters. So I'm going to write down my ICEs again. And my information, I know that my magnification is times 500. I know that my image size is 4.1 centimeters. And I don't know my actual size. I don't know my real length yet. So I'm then going to do my conversions. Now my 4.1 is in centimeters, and I'm ultimately going to want it in micrometers. So I'm going to do my conversion first. I'm going to convert into millimeters, which is multiplied by 10 to get 41. And then I'm going to convert into micrometers, where I'm going to multiply by 1,000, because there are 1,000 micrometers in 1 millimeter. So we get 41,000 micrometers. Next, I write out my equation, image size equals actual size times magnification, and then I substitute in my values. So 41,000 equals the actual size times 500. I solve my equation. My actual size is equal to 41,000 divided by 500, and our final answer comes out as 82 micrometers. It's really important that you show your working in an exam. If you don't show your working and just write the answer, if you make a mistake at some point, the examiner will not be able to give you any other marks. They would just mark your answer as wrong and you will get 0. When you show your working, lay it out in a really clear and easy to read way, like I have done here, because that makes your examiner's life easier to go back and notice, where did you make the mistake? Are they still able to give you a mark? Or in a longer calculation question, are they still able to give you 2 or maybe even 3 or 4 marks perhaps? Make every mark count in your exam. The second part of Module 1 is about the cell cycle and mitosis and cell division. So, the cell cycle describes the life cycle of a single cell, from after the cell has been produced, through the cell division, through to when the cell then divides again. Mitosis is an example of cell division. Lots of students confuse mitosis with the cell cycle. Remember, the cell cycle comes first, is the preparation for mitosis. Now, mitosis, or this cell division, is needed for growth and repair. It is needed to produce new cells so that we can repair and replace tissues. The cell cycle is made up of 3 stages. In stage 1 of the cell cycle, the cell is carrying out its normal activity. When it is carrying out that normal activity, it is also growing. And it is also going to start preparing for the division. It is going to replicate the DNA. It's going to double the amount of DNA within the cell. It's also going to replicate and produce more organelles. It's going to increase the number of mitochondria, perhaps. In stage 2, this is the stage of mitosis. This is when the nucleus divides. But what about cells that don't have a nucleus, I hear you ask? This is our other type of cell division. This is binary fission. Binary fission is cell division in prokaryotes. Stage 3 comes after mitosis, and this is the final part of the division. This part we call cytokinesis. Cytokinesis is when the cell membrane and the cytoplasm divides. And after this, we have then produced two new daughter cells. Two new daughter cells produced. They are always referred to as daughter cells, even if the individual is biologically male. These two new daughter cells, they are also genetically identical to each other. Remember how that DNA was replicated before the division. It means that each of those daughter cells will get one copy of the DNA, and that means that they are identical. Let's go through this process of mitosis in a little bit more detail. So, mitosis starts with the nucleus breaking down. Once the nucleus breaks down, the chromosomes are able to line up on the equator of the cell. Now, the chromosomes, these are the coiled structures of the DNA. For example, in humans, we have 23 pairs of chromosomes, or 46 in total. Once these chromosomes line up on the equator of the cell, the chromosomes are then pulled apart. We call each part of the chromosome a chromatid. So this here, this X structure, this is a chromosome. And on the right here, one part of that chromosome we call a chromatid. So the chromatids separate to each pole of the cell. These chromatids are identical to each other. They have been produced by the DNA replicating. Once the chromatids move to each pole of the cell, then the nucleus reforms around the DNA. Now, these cells that are dividing, these are what we would call unspecialized cells, or we might refer to them as stem cells. A stem cell is an unspecialized cell, or we could say an undifferentiated cell. So it is undifferentiated cell that is not specialized. It does not yet have any of those specialized adaptations that we looked at earlier. Now, the stem cells, when they differentiate, that is when they become specialized. And stem cells are able to produce different types of cells. So differentiation, this is when the stem cell becomes specialized. There are two main types of stem cell that we know about for GCSEs. We have our embryonic stem cells and we have adult stem cells. Now, embryonic stem cells, they are produced in the embryo. Embryonic stem cells, they go on to produce any cell within the body. So we could say that they have a greater power to differentiate. They can form any type of cell. Whereas adult stem cells, they are within the body once it's already been formed. Even children have adult stem cells. Any stage of development that is not the embryo, they would be referred to as adult stem cells. A common place where we might find adult stem cells is within the bone marrow or perhaps within the retina of the eye. These cells are only able to produce certain types of cell. For example, the cells in the bone marrow can only produce the cells that form the blood. This means that they have less of a power to differentiate. Stem cells can also be used for medical treatments. Stem cells could be used to replace any damaged cells that have been damaged by a disease, perhaps. Now, there's different types of stem cells can be used in different ways. Let's take a look at the advantages and disadvantages of these different types of cell. In an exam question, quite frequently you might be asked to evaluate these different types of stem cells. And in an evaluation, you need to do 3 different things. You need to give the advantages of each of the things you are considering, each of the things you are evaluating, and you would need to give the disadvantages of each as well. And then the third thing you would need to do is give a conclusion. So, embryonic stem cells. What are the advantages of the embryonic stem cells? Well, as I say earlier, they can differentiate into any type of cell. They can differentiate into any type of cell. The advantages of the adult stem cell, though, the adult stem cells, there are fewer ethical issues or ethical concerns. The reason why is because the adult can consent to their stem cells being used, whereas an embryo would not be able to consent. The use of the adult stem cells also means that the embryo is not being destroyed. In addition to that, adult stem cells also have additional advantages as well. Adult stem cell therapies are quite well established and quite well proven. We know that they work. And if the patient's own stem cells are being used, then there's no risk of rejection. Another advantage of the embryonic stem cells, though, is that they divide more rapidly, which means we can produce those cells faster. Let's think about the disadvantages for each of those then. Well, the disadvantages of the embryonic stem cells are that there are ethical concerns. The embryo is destroyed. And the embryo cannot consent. In your exam, it's important that you stick to some of this phrasing. Some students get distracted and start talking about how the embryo is a baby or the embryo is alive, and you won't get marks for that. You need to say the phrase that the embryo is destroyed. That is how you get the mark in the exam. In addition to that, other disadvantages are that there is a potential risk of rejection. The patient will recognize those embryonic stem cells as being non-self, i.e. not part of their own body, and that would trigger an immune response. To counteract this, the patient would have to take immunosuppressants. And immunosuppressants, as the name suggests, suppress the immune system. It weakens the immune system. Other disadvantages are because of that rapid division, they can also produce tumours. What about the adult stem cells then? Well, the disadvantages of the adult stem cells is they can only differentiate into certain types, which means we can only treat certain diseases. So can only treat certain diseases. In addition to that, the slower growth rate means that it is harder to produce them. So when we give our evaluation, when we answer an evaluate question, we need to give a conclusion at the end of our answer. And our conclusion is very, very simple. All we need to do is say that ... we choose one of them, embryonic or adult stem cells, is good or bad or better. And then we have to give a reason, because. And when we give a reason, we just need to restate one of the points we've already made in our answer. So for example, we might say embryonic stem cells are better because they can differentiate into any type of cell. Or I could say adult stem cells are better because there are fewer ethical concerns. I need to justify my conclusion. So the second required practical in Module 1 is about growing cultures of bacteria. Now, this could be used to investigate the effects of antiseptics or antibiotics on bacterial growth. We need to use agar plates in order to be able to do this. Now, the agar plate is the nutrient medium that the bacteria or other microorganisms like fungi perhaps are going to grow on. Now, for the bacteria to be able to grow and reproduce, they need that source of nutrients. Now, in an exam, it wouldn't be good enough to just say the word nutrients. So we would need to say that the agar provides a source of glucose, perhaps, or amino acids, as well as important ions, and also a source of water. Now, when those bacteria reproduce, they are going to divide. And the bacterial division, remember, is binary fission. It is not mitosis because it is not nuclear division, because the bacteria do not have a nucleus. Instead, we call it binary fission. The loop of DNA is replicated and then the cell divides to produce two identical cells. So we are going to go from one cell to producing two, to then producing 4, to then producing 8, 16, and so on. Now, when we are carrying out these experiments, we need to use aseptic technique. Aseptic technique is used to prevent contamination. It's vital that the uncontaminated cultures of microorganisms are grown in the lab. If we have the presence of any competing species, this could affect the growth of the cultures, and this would affect the validity of the study performed on them, because we wouldn't be able to determine whether any reduction in the growth of bacteria was due to the antiseptic or due to the antibiotic. There's a range of aseptic techniques that we need to know about. Our first example is working next to a Bunsen burner. This creates a convection current of air. This moves other microorganisms that might be in the air away from the area we are working, and this prevents contamination. Another example of an aseptic technique is sterilizing the apparatus. We're going to use high temperature and pressure in something called an autoclave, which is going to kill any microorganisms that might be on the equipment. We can also disinfect the area. When we disinfect the area, this is going to kill any microorganisms that are on the work surface. We can also flame the inoculating loop. Now, the inoculating loop is the piece of apparatus that we would use to introduce the culture of bacteria onto our agar plate. Flaming the inoculating loop means putting that loop of wire through the flame of the Bunsen burner. What this is going to do is this is going to kill any microorganisms on the loop that may have come from the air after it's been sterilized. We could also flame the neck of the culture bottle. We could open the lid of the culture bottle and pass the neck of that bottle through the Bunsen burner flame. What this is going to do is again create a convection current around the neck of the bottle, and that's going to move the microorganisms in the air away from the bottle. We also want to make sure that we're opening the lid of the Petri dish a very, very small amount, as this is going to minimize the chance of unwanted microorganisms getting onto the plate. We also want to tape the lid at intervals instead of sealing it completely shut. This allows some oxygen in, preventing anaerobic conditions, and that is going to prevent the growth of pathogenic bacteria, which are adapted to grow in the anaerobic conditions. And also we are going to incubate the plates at 25 degrees. This is lower than human body temperature, and this means that any bacteria that might be pathogenic to humans that would be adapted to grow at human body temperature, well, the growth of them is going to be reduced because it is not at their optimum temperature. Instead, we are using a lower temperature. Now, how do we test the effectiveness of these antibiotics then? Well, we need to calculate something called the zone of inhibition. The zone of inhibition refers to the clear circular area surrounding an antibiotic or antiseptic disc on an agar plate where the bacteria have not grown. The area indicates that the antimicrobial substance has effectively prevented the bacterial growth in that region. The size of the zone of inhibition is often measured to assess and compare the effectiveness of different antibiotics or antiseptics against specific bacteria. The larger the zone, the more effective the substance is at inhibiting the bacterial growth. So for example, in our agar plate underneath, we can see we have 4 different regions, A, B, C, and D. Which is the most effective? It would be C. And this is because antibiotic C has the largest zone of inhibition. As well as that, we can see that antibiotic D has no zone of inhibition. That might mean that the bacteria are resistant to antibiotic D. D could also be a control experiment. What this means is that there would be no antibiotic. D is just a filter paper disc and perhaps some distilled water. So the control experiment, there is no antibiotic. And what it does is it shows that antibiotics have caused the zone of inhibition. So it shows that the antibiotic has caused the zone of inhibition. Now, as well as that, we need to control other variables that might affect the growth of the bacteria. Imagine if this was being done on different agar plates with a different antibiotic added to each plate. Well, we would need to control the concentration of the antibiotic. We want to make sure that they all are the same. As well as the concentration, we would also need to control the volume of the antibiotic. We would also need to control the concentration of other nutrients available. So the concentration of glucose, amino acids, other nutrients within the agar. We would also need to control the temperature and make sure that the temperature is the same for each plate that we are growing. And we would also need to control the concentration of bacteria, as if there were more bacteria, there could be more competition and therefore reduced growth. So how do we calculate the population size? So if a bacteria has a mean division time of 32 minutes and has been dividing for 5 hours, how many bacteria will there be? Well, we need to start by working out how many generations there have been. So if in 5 hours it divides every 32 minutes, there is going to be, we're going to convert our hours into minutes. So we're going to do 5 times 60 to get 300 minutes. And then I'm going to divide by 32. And we get 9.375 generations. So we can call that 9 complete generations. Now, after 9 complete generations, because the 10th one hasn't been completed, how many bacteria will there be? Well, this is an example of exponential growth. The number is doubling every single time. So we can use 2 to the power of n, where n is equal to the number of generations. So 2 to the power of n would be 2 to the power of 9 in this example, which is going to be equal to 512 bacteria. Another way we could work this out is we could just go through and just double it each time and double it for 9 times to work out the total number. Our third part of Module 1 is about cell transport. So we're talking diffusion, osmosis, and active transport. Let's break down diffusion to begin with. So diffusion is our net movement of particles from a high concentration to a low concentration. Particles are moving randomly all the time because of the kinetic energy of the particles. And if we have an area where they are more concentrated, over time, they are going to spread out to be less concentrated. Now, if we have a cell in between there, if the concentration is higher outside of the cell, more of the particles are going to be spreading out into the cell than the other way around, the particles spreading from the low concentration to the high concentration. This is why we talk about it as the net movement, because the movement of the particles is still occurring in both directions. It's not just happening from the high to the low, but overall, the number of particles moving from high to low concentration is greater. So what affects the rate of diffusion? Well, diffusion is due to the kinetic energy of the particles. So the temperature is going to affect the rate of diffusion, because the temperature, if we have an increase in temperature, that means we have more kinetic energy. And if there's more kinetic energy, that means that there's going to be a faster rate of movement of the particles and so faster diffusion. Other factors that affect diffusion would be the diffusion distance. How far those particles are moving, whether they are moving through multiple cells in order to be transferred from one part of the body to the other. If the diffusion distance decreases, that means that the particles don't move as far. So the particles don't move as far, and so that means that the rate of diffusion will be higher. As well as that, we have our surface area. The surface area refers to the total area that is available for particles to diffuse in and out of the cell. The greater the surface area, the greater the rate of diffusion. And we also have concentration gradient. Now, concentration gradient is a fancy term for the difference in concentration between two areas. If there is a greater difference in concentration, then that means that the particles are going to spread out faster. Imagine a little bit like riding a bike down a hill. If you've got a steeper hill, that means the bike is going to go faster down that hill. We can think of a larger difference in concentration as being a steeper concentration gradient, and so a faster rate of diffusion. So if our concentration gradient is greater, we're going to get an increased rate of diffusion as well. Examples of substances that might diffuse into and out of cells, we could think of things like oxygen and carbon dioxide diffusing in and out of cells, as well as glucose and amino acids diffusing in and out of cells as well. So different parts of organisms are adapted to try and maximize this rate of diffusion. Let's talk about some adaptations for gas exchange. So gas exchange is where oxygen and carbon dioxide are being exchanged in and out of the organism. Our first example here is at the alveoli. Alveoli are found in mammalian lungs, and in those mammalian lungs, oxygen is being absorbed, is diffusing from the air into the blood, and carbon dioxide is diffusing from the blood into the air in the alveoli, which will then be breathed out. So as the oxygen diffuses into the blood and the carbon dioxide diffuses out of the blood, there is a range of adaptations that are going to maximize this rate of transport. Our first adaptation is the surface area. The alveoli overall have a very, very large surface area. And the consequence of this is it increases the rate of diffusion. As well as having a large surface area, they also have many capillaries, or we could say a good blood supply. The reason for that is that means that the blood is being transported away from the alveoli very, very rapidly. So as soon as the oxygen is absorbed, it is transported away, which means that oxygen depleted blood, or blood that has a low concentration of oxygen, is constantly surrounding the alveoli, and that maintains the concentration gradient. So we can say that it maintains the concentration gradient, and that is going to increase the rate of diffusion. This is how we get the marks in the exam, by saying maintaining the concentration gradient. Our next adaptation refers to the walls of these capillaries and the walls of the alveoli. We can say that the walls, or perhaps we could say the exchange surface, are 1 cell thick. The mistake that students frequently make is they say that the membrane is thin or the membrane isn't thick. And the problem there is it kind of shows maybe a little bit of a misunderstanding, because the cell membrane doesn't change in thickness. The cell membrane stays the same. We want to say that the walls or the exchange surface are 1 cell thick. And the consequence of this is this reduces the diffusion distance. And if we reduce the diffusion distance, the distance that the oxygen has to move from the alveoli into the blood, this is going to increase the rate of diffusion. What about in fish? Now, fish don't have lungs. Fish have gills instead. So the adaptation of the gills to increase gas exchange are very, very similar to the adaptations of the alveoli. So firstly, there is a large surface area as well. This is going to maximize that rate of diffusion. Things like the gill filaments are going to increase that surface area. As well as having a large surface area, there are also lots of blood vessels within the gills. We could say there are many capillaries, or a rich blood supply, and that is to maintain the concentration gradient. And in addition to that, we could also say that the exchange surface, again, is 1 cell thick. What about leaves then? Leaves need to exchange gas with their surroundings. They need to absorb carbon dioxide for photosynthesis, and they also need to absorb oxygen for respiration. During the day, the plant is producing oxygen by photosynthesis, but during the night, it needs to absorb that oxygen from the atmosphere. So our plant leaf also needs to be adapted for gas exchange. Now, we have a part of our plant leaf here called the spongy mesophyll. The spongy mesophyll is where the majority of the gas exchange is taking place, and we can see that it has lots of air spaces. And by having lots of these air spaces, it also increases the surface area, and that is going to increase our rate of diffusion. We also have the stomata and the guard cells. Now, the stomata and guard cells control the movement of gases in and out of the leaf. Let's now move on to osmosis. Osmosis is a special case of diffusion. Osmosis we can define as the net movement of water from a dilute area to a concentrated area through a partially permeable membrane. Let's break down this definition. So like we said with diffusion, we're talking about the net movement. It is the movement in both directions, but overall, the water is moving from the dilute to the concentrated area. Osmosis is specific to water. We can say that water moves by osmosis, and in an exam question, we should always say that because that's how we secure and make sure that we always get the marks. Now, this phrasing of dilute area to a concentrated area. Now, sometimes people might say osmosis is the net movement of water from a high concentration to a low concentration. But what are you talking about? Are you talking about the concentration of water? Are you talking about the concentration of the solute? It's unclear what your definition is referring to. Whereas if we say from a dilute area to a concentrated area, there's no ambiguity. It's obvious what we are saying. So this will make sure that you always get the marks. And then we also say through a partially permeable membrane. An example of a partially permeable membrane would be the cell membrane. It controls what goes in and out of the cell. Some things can pass through it, like water. Other things cannot pass through it. Let's look at an example. So I have two cells here. My first cell, we can see we have a high concentration inside the cell. So inside the cell, we could say that it is concentrated. Outside the cell, we could say that it is dilute. So what is going to happen is the water, the blue area, is going to move into the cell. The water is going to move inside the cell, and what that's going to do is that's going to cause the cell to expand. The cell is going to gain mass. Now, my example underneath, this time round, the concentration inside the cell is less than the concentration outside. So inside the cell is dilute, whereas the outside of the cell is concentrated. And in the second example, water would then leave the cell. And when the water leaves the cell, it is going to lose mass, or it is going to shrink. So, given that osmosis is a specialist case of diffusion, what factors might affect the rate of osmosis? Well, we could talk about temperature again, because as the temperature increases, that is going to increase the kinetic energy of the particles, increasing the rate of the movement and diffusion and osmosis. We could talk about the distance. If the water has to move further, it has to move through more cells, it's going to take longer. We could talk about the surface area. When we talk about the surface area, the greater the surface area, the more area there is for the absorption of water, and so the greater the rate of osmosis. A good example of increasing surface area would be like the root hair cell. And also we have concentration gradient. The greater the difference in concentration, the greater the difference between the dilute area and the concentrated area, the greater the rate of osmosis is going to be. So let's have a look at how osmosis affects cells. On the left-hand side here, we have animal cells. Now, animal cells, as we'll remember, do not have a cell wall. They only have a cell membrane. Let's go through these three examples. So in the first example, we can see we have water moving into the cell and out of the cell. We have zero net movement. That is because we have the same concentration inside and outside. We could refer to this as being isotonic, iso meaning the same. And if there's zero net movement, well, the cell stays the same size. It doesn't gain or lose any mass. Our next example here, we can see that water is moving out of the cell. So the water moves out, and as we say, by osmosis, making sure we say that every time. If the water is moving out of the cell, it's because the area inside the cell must be more dilute than the area outside. So we could say that it is more concentrated outside. And that is why the water is leaving. If the water leaves, that means that the cell is going to shrink. It is going to lose mass. Let's look at our third example then. Now in our third example, we can see that the water is moving in. Now the water is moving in because the outside must be more dilute. There's more water outside compared to inside. And this means that the cell is going to expand. But the problem is animal cells do not have a cell wall, and the cell membrane can't just continue expanding indefinitely. It's a little bit like a balloon. Eventually, the cell might burst. Let's compare this now to the plant cells. Now, the plant cells here on the right-hand side, our first example here, we can see that the cell membrane has pulled away from the cell wall. What has happened is the cell has shrunk. The cell has got smaller. The cell membrane pulls away from the cell wall. This is known as plasmolysis, or we could say that the cell is plasmolysed. The reason for this is because the cell has shrunk. The cell has lost water. So the water moves out. And the reason the water moves out, just like with the animal cell, is because it is more concentrated outside. And then our last example here, our plant cell has expanded. And the reason the plant cell has expanded and that cell membrane is pushing up against the cell wall is because the cell has gained water. We can see the vacuole in the middle here has got bigger. So the cell gains water. And we could describe the cell as being turgid. The reason for this is because it is more dilute outside the cell. The third required practical in Module 1 is the osmosis required practical. Now, in this practical, we are investigating how different solute concentrations affect osmosis into and out of plant tissue. Predominantly, we're going to use potatoes, but any root or any plant cells could be used potentially. Now, what we're going to try and do here is we're going to try and determine the concentration of the plant tissue or concentration inside the cell. Now, in any investigation, we have three different variables: our independent, our dependent, and our control variable. Our independent variable, this is what we are going to be changing in the experiment. And in this experiment, it is the concentration of the solution. The dependent variable is what we are going to measure. And in this investigation, we are going to measure the change in mass. But one of the problems is that we can't make sure that the starting mass is identical for all of our pieces of potato. We can't make sure that they have exactly the same number of cells. So in order to be able to make that comparison, we are going to measure the percentage change in mass. This is so that we can compare the individual pieces of potato. And the control variables are what we are going to keep the same. These are factors that would affect the rate of osmosis, and so we need to keep them to the same to make sure that the only thing that is affecting it is the concentration difference. So this could be things like the temperature. This could be things like the volume of water, making sure that there is an excess of solution or excess of water over the pieces of potato. But also we could think about the surface area or the size of the potato as well. So the size or surface area of the potato. Now, because it may not be possible to make sure the surface area is exactly the same, this is again another reason as to why we are going to calculate the percentage change in mass. Another thing that would affect the rate of osmosis, or another thing that would affect the change in mass, would be the time that we leave each of these pieces of potato for. If we leave them for longer, more osmosis is going to take place. So how do we make sure the experiment is more repeatable? Or how do we improve the reliability of our data? Well, what we are going to need to do is we're going to need to repeat at each concentration. And when we repeat at each concentration, what that is going to do is that is going to improve the precision. As well as repeating, we are also going to calculate a mean. And when we calculate a mean value, we are then going to potentially remove anomalies before we calculate that mean as well. An anomaly is an outlier from the data. It's something that doesn't match the trend or the rest of the data. We also want to improve the validity of our experiment. And to improve our validity, we want to make sure that we are only measuring the mass of the cells themselves. So we might want to remove any excess water or remove any surface water. And by removing that surface water, perhaps by blotting it with a piece of paper towel, what we are going to do is we are going to minimize the error in our experiment. Once we've collected our data, we can then go ahead and calculate our percentage change in mass. Our percentage change in mass is calculated as our change divided by the starting mass multiplied by 100. And you can see that some of our percentage changes are negative, and that tells us that the cells have lost mass. In an exam, you might be asked to plot this data as a graph. To plot your data as a graph, what you need to do is you need to first label your axes. Now, my x-axis here is going to be my independent variable, so it's going to be my concentration, and my units for concentration are moles per decimeter cubed. It is important that we make sure we always give our units in our graph. I also need to provide a suitable scale. Now, your suitable scale should cover at least half of the grid, as well as being sensible. So no 3s, no 6s, no 7s, 8s, or 9s either. So in this instance, we could go up in 0.1. But if I only go up in 0.1 using the smaller squares, I won't occupy half the grid. So I'm going to use two large squares to go to 0.1, and then 0.2, 0.3, and then 0.4. And then I'm going to plot my data. Now, you're going to get 2 marks for plotting the data in your exam, so it's really important that you make sure you do it accurately. Use crosses to plot your data, and use a sharp pencil so that if you make a mistake, you can rub it out and you can redo those crosses. Now, I've plotted my data here, and I can see that we have a curved shape. I can see that it's definitely going to be a curved line of best fit. If the data looks like a straight line, it's probably going to be a straight line. If it looks like a curve, it's probably going to be a curve. So when you do your curved line of best fit, again with your pencil, making sure you do a single smooth line, like so, connecting the dots together, but in a single smooth curve. And then we need to determine what the concentration is inside the cell. Now, the concentration inside the potato cells, we would be able to work that out if the concentration inside was the same as the concentration outside, if it was at that isotonic point. Now, if the concentration inside and outside are the same, there is going to be 0 change in mass. And if there's 0 change in mass, well, that would be on our x-axis there. So where is there 0 change in mass? At this point here, the x-intercept. And we can see there, that's at about 0.31. So the concentration inside the cell must be 0.31 moles per decimeter cubed from our graph. We also need to know about active transport. Active transport is the movement of particles against the concentration gradient. Or we could say from low concentration to high concentration. By moving against that concentration gradient, thinking about the concentration gradient as being that hill that we're riding our bikes down, well, if we're going up that hill, this is going to require energy. And if we're requiring energy, we're going to need respiration in order to provide that energy. So what is going to be affecting the rate of active transport? Well, temperature, because if the particles are moving faster, we're going to get a faster rate of active transport. Distance, perhaps. If the particles have to move further, we have a faster rate of active transport. Surface area will be affecting the rate of active transport as well. But additionally, we also have the rate of respiration. If more respiration is taking place, that means that we have more energy available to carry out active transport. Examples of active transport would be things like the absorption of glucose in the small intestine or the absorption of mineral ions by the root hair cells in the soil. And there we have it, Module 1 completed. Give this video a like, make sure you subscribe as well and click the bell because I'm going to be breaking down all of the other modules too.